All right, so our group is Emma, James C, Melinda, Rebecca, myself, and Simon. And today's talk is on autosomal dominant polycystic kidney disease and the potential use of CRISPR as a tool in treatment and potential prevention of disease.
So autosomal dominant PKD is the most common monogenic kidney disorder. It has an incidence rate of 1 in 500 to 1 in 1,000, affecting up to 250,000 Australians and 7.2 million people globally. ADPKD has nearly complete penetrance, and almost 100% of those with the mutation develop renal cysts, many of which progress to renal insufficiency. However, disease severity exists on a spectrum. ADPKD is primarily characterized by mutations in 2 genes, PKD1, located on chromosome 16, shown here, and PKD2, located on chromosome 4. 85% of cases are associated with mutations of PKD1, whilst only 15% are associated with PKD2, with mutations to PKD1 typically resulting in a more severe disease onset. As such, the focus of our Cas3 approach is on the PKD1 mutation due to its increased incidence rate and severity. The PKD1 gene encodes for the transmembrane protein receptor, polycystin-1, that in conjunction with polycystin-2, the protein product of the PKD2 gene, forms a receptor channel complex. This complex localizes to the primary cilia on the apical surface of epithelial cells, as well as sites of cellular attachment. The PC complex is thought to release intracellular calcium stores and regulate multiple signaling cascades, particularly those associated with regulating cell proliferation, fluid secretion, and tubular morphology. Mutations to the PKD1 gene result in reduced activity of the PC1, leading to dysregulation of calcium signaling and subsequently disrupting normal cellular activity that maintains cell structure and growth. This is evident in the cystic phenotype.
The defining characteristic of polycystic kidney disease is the presence of cysts in both kidneys. Cysts are fluid-filled sacs which grow in size and number over time. As the cells proliferate and increase in size, they will begin to disrupt normal renal function, eventually resulting in end-stage renal disease. Other symptoms associated include kidney stones, which develop in 20 to 30% of patients and are frequently associated with the acute onset of hematuria, pain, and rapid kidney deterioration. Other symptoms related to the kidney include urinary tract infections, cyst infections, polyuria, and hematuria. Other symptoms include hypertension, which is the most common symptom and can be found within approximately 60% of ADPKD patients before extensive cyst growth. The flowchart on the slide here shows the pathogenesis of hypertension in ADPKD, and it highlights the importance of the RAS pathway in hypertension development. As cysts increase in size, they compress the surrounding renal vasculature, triggering the activation of the RAS pathway, which leads to hypertension. Hypertension, if untreated, will reduce the blood flow to the kidney, thereby increasing the amount of kidney damage which occurs, perpetuating the cycle. Extra-renal cyst development occurs in approximately 60% of individuals. Generally, it's an incidental finding, but women are more likely to develop severe hepatic cysts. And one of the most severe extra-renal results of ADPKD is intracranial aneurysms, which have been noted in up to 40% of patients. These aneurysms rupture, causing intracranial hemorrhage and death in 10% of patients.
Whilst there is currently no cure for ADPKD, some treatments include kidney transplants and renal dialysis, both of which aim to compensate for renal failure. However, neither address the extra-renal manifestations of the disease, and as such are more end-stage treatments rather than preventative measures. Diet and lifestyle are important ways of managing the associated symptoms of ADPKD. By reducing alcohol, caffeine, sodium, and protein consumption, less strain is placed on kidney function. Tolvaptan is a new pharmaceutical treatment that blocks vasopressin receptors, preventing fluid accumulation in the cysts. Whilst it has been shown to slow the growth of kidney cysts, it is only recommended for patients with rapidly progressing disease and has adverse side effects associated, such as polyuria and liver enzyme disruption.
We are proposing a CRISPR-based gene editing strategy for treatment of ADPKD. CRISPR, originally developed from our bacteria, utilized CRISPR arrays and Cas proteins as a form of viral immunity to target and destroy invaders' DNA. The Crispr method uses this to target specific DNA regions with guide RNAs complementary to the target strand. Using Cas9, dsDNA is cleaved, and with Cas3, many nucleotides are removed. The strand repairs itself with mechanisms like homology-directed repair, and this is manipulated to create genetic modifications at desired locations with high specificity and efficiency, and it can be used in a variety of cell types. The mouse PKD1 gene is very similar to the human form, so we can test in mice before having human trials. There are a number of benefits to initially testing in the murine model instead of testing in humans, because mice have shorter generation times, so they can grow many mice and get them to express the disease quicker than in humans, improving the accuracy and reliability of our research. We can check that our strategy will be effective before doing human trials. We can also find out if it will cause problems in other organs or any other unwanted effects. The use of mice also presents less ethical barriers for initial research and testing of our approach. We will look at targeting murine models homozygous with this specific PKD1 allele. The molecular lesion is a T to A substitution, changing an isoleucine residue to an asparagine. This involves the homolog of the human PKD1 gene and closely mimics the monogenic nature of ADPKD. Additionally, embryos are readily purchasable from the Jackson Laboratory.
There have been many advances to create accurate single nucleotide repairs. For example, nickase Cas9 fused with cytidine deaminase has been shown to be able to generate single nucleotide substitutions without DNA deletions. While this would be an exemplary application to this allele, as well as many somatic and germline human mutations, we wanted our approach to comprehensively target all mutations that create ADPKD pathogenesis. Here, our approach is as follows. We aim to use a CRISPR-Cas3 system to remove the entire faulty gene and via homology-directed repair, insert the new gene. Here, we are using a precessive nuclease Cas3 system together with a minimal type 1C cascade-based system for targeted deletions. In general, most gene editing techniques have employed single subunit class 2 CRISPR-Cas systems, for example, Cas9 and Cas12a, but they are limited in their ability to create large deletions. As the region we are targeting is well over 10 kilobases, we can use a class 1 type 1 system, Cas3, which recent studies have shown to processively remove between 7 to 424 kilobases. Unlike Cas9, where it will create a double-stranded break at a designated point in the genome, it will shed away sequences, ensuring they will not be reintegrated back into the sequence. As this study has also shown a success in allowing for homology-directed repair in humans, we have opted for this approach. To begin with, the type 1C CRISPR-Cas system used in gene editing involves 4 proteins. First, Cas5, Cas8, and Cas7 proteins come together to form the CRISPR RNA-guided cascade surveillance complex. This, as shown here in the slides, contains a guide RNA or gRNA, which contains the scaffold sequence necessary for Cas binding, the double-stranded section, and the user-defined 20 to 32 nucleotide spacer that defines the genomic target to be modified. The DNA should be complementary to the spacer, and to find this match, the cascade first searches for a protospacer adjacent motif, or PAM sequence. They are generally NGG in structure, and once there is a match, the cascade will check if the gRNA has any complementarity with the DNA strand. If there is, it will form a stable R loop and recruit Cas3 to begin its helicase nuclease activity. From there, Cas3 will begin to degrade the DNA. The cascade creates a nick upstream of the 5' PAM sequence, and Cas3 has ATP-dependent 3' to 5' translocase activities to degrade the single-stranded DNA target. Now, in terms of our murine model, we have designed it so that we have 2 type 1C systems that target both strands of the PKD1 gene. Complementary gRNA have been generated and will effectively remove the gene once added to pluripotent mouse cells. Also, in choosing our gRNA, complementary gRNA has been generated using ChopChop, and we're chosen to cover both strands as well as cover the entire gene, as shown. Now from here, we will aim to insert the wild-type non-pathogenic variant of the PKD1 gene into the murine model. In general, mammals repair double-stranded breaks in the DNA via non-homologous end joining, NHEJ, or homology-directed repair, HDR. We will employ the use of HDR given its accuracy to incorporate DNA inserts into the genome. While HDR can only be used by the cell when there is a homologous piece of DNA present in the nucleus, which is mostly in the G2 and S phase of the cell cycle, this is fine, since we are dealing with stem cells which are not senescent and do divide. The first step is to generate a DNA repair template. The DNA repair template is made up of the region of DNA that you want to insert, as well as homology arms. These homology arms are used to help direct our insert into the deleted region, so these arms should be complementary to the DNA regions flanking the left and the right side of the deletion. For this murine model, with homology arms of around 2 kilobases and a transcript of around 14 kilobases, GenBrick building blocks of this length can be purchased from GeneScript for around $10,000. The next stage is to transfect or insert the DNA repair template into the mouse stem cells. The cells will uptake these templates, and as the homology arms bond with the complementary sections of the genome, the DNA will be incorporated through homologous recombination. To increase the rate of HDR, as in mammalian cells, NHEJ use far outweighs HDR. We will insert a variant of the E3 ubiquitin ligase called RAD18, variant E18. Studies have shown that supplying RAD18E18 can inhibit NHEJ by binding to the p53 binding protein 1, the latter being necessary for NHEJs. Thus, this will ensure a higher success rate of stem cells with the amended gene.
These cells will need to target are renal epithelia, which are the cells lining the tubules and ducts of the kidneys. Studies have shown that viable renal epithelia can be collected from urine samples, which can then be grown in culture. The cells can be cultured on any standard growth medium. This provides a non-invasive means of collecting the cells without further damaging the affected kidneys. Once grown in culture, these cells may be induced back to partial pluripotency using the reprogramming factor technique set out by Roney et al. The DNA editing tools can then be added to the cells using microinjection. This technique will be used to safely deliver a comparatively large piece of DNA into the cells. Once inside the cells, the treatment would correct the PKD1 gene, as we mentioned earlier. The DNA of the treated cells would be repaired using the cell's natural mechanism of HDR. These treated cells would then be injected back into the study animal, in this case a mouse, via the renal artery. In theory, the same method could be used to deliver treatment to other affected organs, but we'll discuss that further when we talk about possible future directions. For now, the treatment would be exclusively supplied to the kidneys. Once travelling through the renal passages, the stem cells will be able to establish new colonies. The significance of the induced pluripotency is that it allows the reintroduced treatment cells to re-establish normal renal architecture as they differentiate back into normal epithelial cells. Thus, the mouse would end up with newly functional renal epithelia, which would counteract the dysfunctional cells. Although other somatic cells could be induced with pluripotency for the same purpose, using cells which had been epithelial cells previously simplifies the process of inducing pluripotency, and so means it's less likely to go wrong.
We've laid out the pros and cons of our strategy using a mind map. However, the biggest considerations for our approach are whether or not it will be a viable treatment for ADPKD in humans, which we can only determine through replication, how long the potential treatment timeframe is. Our gene has a pro because it has many PAM sequences both upstream and downstream, so it is easy to find sequences for the GNRA spaces. Potential issues include issues of crossover between mice and humans, the potential steric hindrance of us using a larger Cas3 nuclease versus a Cas9, and the risk of off-target effects, which many possible off-target effect sites have already been identified through CHOP-CHOP. While we are not conducting our initial trial on humans, there are still many ethical considerations we need to take into account. The primary ones for the murine model are the risk of off-target effects of the Crispr and to potentially worsen the phenotypic modification of ADPKD. When eventually adapted for human cells, some more ethical considerations include, again, the potential worsening of the disease if transition from mice to humans is unsuccessful, and the ethics of germline modification and the creation of GMO humans, and again, issues of off-target effects and getting approval for treatment.
Although we believe this strategy will be effective at treating PKD, we also feel that this research could open doors to further progress. A key drawback of our proposed technique is that it's a treatment rather than a cure. In addition to meaning that treatment would probably have to be ongoing, this technique would likely also have minimal impact on cysts forming away from the kidneys. Our next stage of research will therefore be to examine if this technique will work if supplied elsewhere in the body that cysts are formed. If that is successful, we can then start to examine the possibility of applying it to embryos to ensure that all the body's cells express the unmutated PKD1 gene as required. This could demand the use of easy CRISPR to increase the accuracy of HDR in these valuable cells. This would effectively cure PKD1 and make further treatment unnecessary, even for later generations. Either our original technique, or if laws permitted, our embryonic technique, could then be tested in progressively closer related species, with the end goal being to one day test in humans.
Here we've provided the references for all the sources used. Each reference has been grouped according to where it first appears in the presentation. So that wraps up our presentation. We hope you all learned something new about PKD and the potential applications of CRISPR in the treatment of PKD. Are there any questions?